Good day, everyone. For today's topic, I will be teaching you how to properly process your tissues in the histopath lab. But in order for you to have a grasp of this subject, you need to know them by heart first before getting into details. So today, we will just be tackling the overview of the different steps in tissue processing. But before that, I want to review the simplified meaning of our subject, histopathologic and cytologic techniques with general pathology. When you say histopathologic, that means the study of the different diseases and pathologic processes that is found in the tissues. Histo, patho, and logi or logos. When you say cytology, the study of cells, and what are the techniques used in order for you to assess the pathologic processes that is happening in the cells and in the tissue. So is that easier for you to understand the subject of our topic? So that you will know the meaning instead of memorizing the entire thing. Do you get me? Let's proceed. Fresh tissues are usually examined when there is an immediate need for evaluation. A better and more effective means, however, of studying tissues, whether normal or abnormal, is by examination of their sections and smears which have been permanently preserved, stained for demonstration of specific structures, and mounted on glass slides with cover slips for permanent keeping. The one that you can see in your human anatomy and histology laboratories. Can you remember that? Those slides are created in the histopath lab. Solid structures and tissues must be preserved and carefully processed in this following order. Let's start first with the first and most critical step in histotechnology involving the fixing, or otherwise known as the preserving of the fresh tissue for examination. This process class is called fixation. The quality of the section on the slide is only as good as the quality of the fixed tissue specimen. So if you properly fix your specimen, then you will get an optimum result for the pathologist to be read. The primary aim of fixation class, this is to preserve the morphology and the chemical integrity of the cell in as lifelike a manner as possible. So whatever is found in your body, with the help of fixation, this will be reflected in the slide if you properly fix your specimen. The cell, of course, is composed of what? Proteins, carbohydrates, lipids, and other organic and inorganic substances. Fixation class prevents degeneration, decomposition, putrefaction, and distortion of tissues after removal from the body. The most common fixative that we use in the laboratory in Cebu Doctors University Hospital is the neutral buffered formaldehyde, or otherwise known as NBF, neutral buffered formalin. So kindly write that down. So the most common fixative that is used in our hospital is the NBF, the neutral buffered formaldehyde or formalin. So again, what is the purpose of fixation? This is to preserve the specimen in as lifelike as possible. As soon as tissue has been fixed and the bones and teeth have been decalcified, it is necessary to remove the fixative and water from the tissue and replace them with a dehydrating fluid in preparation for impregnation. The process of removing the intercellular and extracellular water from the tissue following fixation and before wax impregnation is what we call dehydration. So, the dehydrating agents are usually in increasing strengths to remove aqueous tissue fluids with little disruption to the tissue caused by diffusion currents. So in the histopath lab in CDUH, we usually start with 60% alcohol, 75% alcohol, and then 90% alcohol going to methanol. So the most common dehydrating agent that we use in the histopath lab is ethyl alcohol. So we start from a smaller percentage or concentration going to a higher or increasing strength of alcohol concentration. As a general rule, class, you write this down. As a general rule, whatever dehydrating agent that is used, the amount in each stage should not be less than 10 times the volume of the tissue. This is in order to ensure complete penetration of the tissue by the dehydrating solution. So your specimen must be fully submerged with the dehydrating fluid as well as your fixative. You remember that your fixative must be more than more than your specimen. Step is clearing, or what we call de-alcoholization. De-alcoholization meaning to remove alcohol. And where did you get the alcohol? From the dehydration step. So this is the process whereby alcohol or a dehydrating agent is removed from the tissue and replaced with a substance that will dissolve the wax later on, with which the tissue is to be impregnated. So this clearing agent will give the tissue a translucent appearance, meaning that's why we use the term clearing. So to remove alcohol, to remove dehydrating agent, and change it with a substance that will give it a translucent appearance. When used after the tissue section has been stained, the clearing agent will make microscopic tissue preparations transparent due to their high index of refraction. The most common clearing agent that is used in the laboratories is silene. So the clearing agent is silene. So you already know three agents, right? For fixative, we have formalin. For dehydrating agent, we have ethyl alcohol in increasing grades. And for clearing agent, we have silene. Next step is infiltration, or otherwise known as impregnation. This is a process whereby the clearing agent is completely removed from the tissue and replaced by a medium that will completely fill all the tissue cavities, thereby giving a firm consistency to the specimen and allowing easier handling and cutting of suitably thin sections without any damage or distortion to the tissue and its cellular components. The most common and the simplest tissue infiltration media is the paraffin wax. Kindly write that down. Paraffin wax is the simplest, most common, and after impregnation, the tissue is placed into a mold containing the embedding medium, and this medium is allowed to solidify. So your paraffin wax tissue already is placed in a medium or a mold. Can you imagine in your refrigerator the one that has ice trays? So your embedding medium looks like that. Okay? But by the way, one example of an embedding media is your ice trays. A very, very nice example, no? So your paraffin embedded tissues, so from your infiltrating medium, they're arranged at the bottom of the mold together with their proper labels and immersed in melted paraffin at a certain temperature. You call this technique as orientation. So you write this down. Orientation is the process by which a tissue is arranged in precise positions in the mold during embedding, on the microtome before cutting, and on the slide before staining. So this technique is what we call orientation. Before you subject your paraffin-embedded tissues to section cutting or microatomy, you must make sure that you properly trim the edges of the paraffin-embedded tissues to remove the excess wax. The seventh step in tissue processing is what we call section cutting or microatomy. The process by which processed tissue, most commonly a paraffin-embedded tissue, is trimmed and cut into uniformly thin slices or sections to facilitate studies under the microscope is what we commonly known as microatomy. The basic instrument used is a microtome that is capable of cutting a section at a predetermined thickness around 4 to 6 micra. So you write this down, please. 4 to 6 micra with a steel knife or a blade. Remember that in your risk management lecture, microatomy or section cutting is the most common accident that can happen in the histopath laboratory. So a medical technologist must be very, very, very careful in handling the paraffin-embedded tissues while around the microtome. Step number 8, that is staining. So this is the process of applying dyes on the sections to see and study the architectural pattern of the tissue and the physical characteristics of the cells. This is made possible because different tissues and cells display varying affinities for most dyes and stains used during this process so that they become more visible. So we will tackle more about staining on your staining lecture. So for now, just know that during staining, this is the application of the dyes to your paraffin embedded tissues that is already section cut and now prepared on a slide. The most common staining technique that we use in the histopath lab is H and E staining. H and E staining. The second to the last step is a step wherein the main purpose is to physically protect the section and preserve the slides for permanent keeping. This technique is what we call mounting. So if you see in your slide, it is properly stained, properly oriented, then this slide is already very ready for mounting. Mounting is when you place a cover slip over the top of the stained tissue section that is on the slide already. Just place 3 drops of mounting media and you make sure to properly orient also your cover slip on top. And then just close it. Let the syrupy fluid run all throughout the sides of the cover slip, and that is ready for reading by the pathologist. The most common mounting media used in the histopath lab in CDUH is what we call U-kit. EUKITT. This is the most common mounting media used in the laboratory for CDUH. The last but not the least is, of course, one of the most important steps is labeling. You have to make sure that you properly label the tissue section from fixation part until this labeling step that is ready for reading by the pathologist. Any problems within the technique or within the process, then for sure the result is also messed up. So you have to make sure that the labels from requisition to fixation down to staining and then mounting and then your labeling part are all the same. So you have to ask me, you can always ask yourself, Miss, how about if the paper during the fixation part will be lost? That is the reason why you always have to label your slides. You label your slides with a pencil. This will keep you from mixing other specimens from your own. This will also help you identify that this slide or this tissue is just for this slide and for this patient and for this case. Just you understand that? So please always make sure to be mindful of the labels from the requisition or the request form down to the labeling process of tissue processing. The video shown a while ago is what we call the Autotechnicon. An Autotechnicon is an automatic tissue processing machine that fixes, dehydrates, clears, and infiltrates tissues, thereby decreasing the time and labor needed during the processing of tissues, resulting in a more rapid diagnosis with lesser technicality. The principle of an Autotechnicon is that it passes tissues through its sequence of solutions by means of a carriage raised by a drive mechanism, which in turn is operated by a clock-controlled timing disc. An Autotechnicon has an electrical clock which controls the time needed for each processing time. So it has an electrical clock as well as a transfer arm, which is used to move from one receptacle basket going to another container and finishing all the 12 steps. The timing disc may be notched as desired, thus allowing for variation as well as accuracy of timing. It only needs 2 to 3 changes of wax for infiltration that is required to remove the clearing agent and properly impregnate the specimen by constant agitation, thus improving tissue penetration, giving rise to more consistent results. This Autotechnicon class has 12 individual processing steps, so there are 12 containers, glass containers, with 10 1-liter capacity glass beakers and 2 thermostically controlled wax baths that is used for paraffin wax impregnation. It also has a safety device cutout switch which protects the wax against overheating. There is a continuation of agitation of the fluid that is also accompanied by a continuous vertical movement or rotation of the specimen carrier by a mechanism connected to the transfer arm. If you can remember from the video that I have shown you a while ago, that is how they agitate the fluid. There is a continuous vertical movement and rotation going up and down from one glass beaker to the other until it finishes all the 12 processing steps. To place tissues in the Autotechnicon for processing, they should be enclosed in small tissue receptacles. So your tissue specimen is placed on those yellow, cute, small tissue receptacles. Can you see that on the screen? And those small tissue receptacles will be placed inside the receptacle basket and attached to the carrying yoke. The receptacle basket is attached back one position from the first solution for processing. The processes involved in Autotechnicon uses a one-basket system. This means that for each process, there is a corresponding basket number that will be given and a corresponding time in each of these baskets. Let's say for fixation, formalin is placed in basket number 1 and basket number 2. The time needed for each is 1 hour. So this means for fixation itself, you have allotted 2 hours for 2 baskets. Let's proceed to dehydration. Dehydration contains an increasing grades of alcohol. Remember that? So this means that in basket number 3 to 5, you have to allot 1.5 hours for each basket. And for basket 6 to 8, you can allot 1 hour for each basket. This means that for basket 3, let's say that's 60% alcohol. For basket 4, that's 75% alcohol. Basket 5, that's 80% alcohol. So that means 1.5 hour each basket. That for 6 to 8, that's only 1 hour each basket. Let's say, for example, for basket 6, that's already 90% alcohol. Basket 7 is 95% alcohol. And lastly, for basket 8, that's 100% ethyl alcohol or already methanol. That's 1 hour for each. Is that understood? Okay. For clearing, this contains the clearing agent xylene. This means in basket 9 and basket 10, you have to allot 1.5 hours for each basket. And lastly, for infiltration, this contains the 2 thermostically controlled wax baths for paraffin wax impregnation for your tissue specimen. This means that you have to allot 2 hours for each basket. Okay, so you have to memorize this by heart. So let's have one example about this. Let's say the time started for tissue processing is at 4 p.m. Okay, 4 p.m. This means in fixation basket number 1, you have to allot 4 to 5 p.m. And then for fixation number 2, since this is another an hour, that's 5 to 6 p.m. So just for fixation, you have allotted 2 hours. That's 4 to 6 p.m. for 2 baskets. This is shortened because of agitation. So with the help of agitation, class, the fixation process is shortened. So for dehydration, that's increasing grades of alcohol. That's 1.5 hours each for baskets 3 to 5. So that's giving you from 6 to 7:30, 7:30 to 9, 9 to 10:30. So you'll be finished by basket 5 at 10:30 p.m. Then continuing dehydrations, basket 6 to 8. That's 1 hour each. So from 10:30 to 11:30, 11:30 to 12:30 a.m., 12:30 to 1:30 a.m. So by 1:30 a.m., you are done with dehydration without human power. This is all the work of the Autotechnicon, properly timed using the electrical clock and with the help of its transfer arm. After the last basket for dehydration, let's proceed to clearing. This gives you another half hour, 1.5 hours for both baskets 9 and 10. So from 1:30 to 4:30 a.m. Lastly, for infiltration or impregnation, that's using paraffin wax, you have to allot 2 hours of each. So from 4:30 a.m. to 8:30 a.m. By the time you go back to the laboratory, your tissues are already fixed, dehydrated, properly cleared, and infiltrated, now ready for embedding. So this is what makes our lives easier in the histopath laboratory with the help of the Autotechnicon. In each laboratory section, there's always QC. The majority of tissue processing is done by the Autotechnicon. Thereby, it reduces the workload of the medical technologists in the lab. However, there are some major drawbacks in this machine, and one is the timing of processing. So it is very important that you set the time and calibrate the machine properly so that it will hit the exact time, and it will not be overly fixed, overly dehydrated, and also not under fixed, dehydrated, cleared, and infiltrated. Thank you so much for listening. I hope you've learned something from me today. For any questions, I will set up a question poll in our Histopathologic page in Facebook so that you'll have at ease and of questioning and asking me, and there will be uniform answers for everyone. Thank you so much and have a great day.